Welcome to Catch This. My name is Trudy Ray. Public health officials are continuing to monitor the spread of avian H5N1, the strain of influenza virus associated with the bird flu. Although there is still no evidence that this virus can transmit from one person to another, several recently infected individuals are attracting close attention because the source of their infection is unclear. I will talk about two of them in this video and what these cases mean for H5N1 in humans. Don't go away. One of the individuals is a child in California, the first documented pediatric case of H5N1 in the United States. The child allegedly had no exposure to any infected animals, and it is unclear where the virus originated. Contact tracing has not revealed any other H5N1 cases in the child's social circle, suggesting that the virus was not acquired from or passed to another human. Fortunately, the illness was mild and the child has recovered. Meanwhile, a more serious H5N1 infection was reported in a British Columbian teenager. As with the child in California, it is unclear how the teen became infected. However, genetic sequencing analysis implies a possible exposure to wild birds. None of the teenager's close contacts were infected with H5N1, which makes person-to-person transmission unlikely and indicates that the virus is not circulating in the community. The teenager's symptoms started with an eye infection, but later developed into a severe lung infection, suggesting that the virus evolved an enhanced ability to enter respiratory tract cells within that individual after it infected that individual. Influenza viruses enter host cells when the viral surface hemagglutinin protein, in other words the H in H5N1, binds a host cell surface protein known as a receptor. Human influenza viruses bind receptors that consist of a sialic acid molecule linked to galactose via an alpha-2,6 glycosidic bond, and these receptors are located mostly on cells of the upper respiratory tract. In contrast, avian influenza viruses, including H5N1, preferentially bind sialic acids with an alpha-2,3 linkage, which are abundant on cells of the avian digestive tract and cells of the human lower respiratory tract. As described in detail in Catch This episode 45, linked above, this receptor preference prevents H5N1 from transmitting efficiently from one person to another. To pass easily among humans, H5N1 would at the very least have to acquire an amino acid change that allows it to bind sialic acid receptors with an alpha-2,6 galactose linkage. A recent study, which analyzed all possible amino acid changes in the influenza virus hemagglutinin that affect alpha-2,6 linked sialic acid usage and cell entry, revealed that some mutations led to enhanced entry of viruses into cells that displayed alpha-2,6 linked sialic acid receptors. In particular, mutations that changed the amino acid glutamate at position 190 of the hemagglutinin protein to a glutamine or alanine, or mutations that changed the amino acid glutamine at position 226 to a leucine or arginine, led to enhanced entry of viruses into cells that had alpha-2,6 linked sialic acid receptors. The published sequence of the H5N1 virus, isolated from the British Columbian teenager, displays nucleotide differences that produce ambiguous amino acid changes at positions 190 and 226 of the hemagglutinin amino acid sequence, meaning that they could be any amino acid. This ambiguity is likely due to the presence of mixed viral sequences and further supports the notion that the virus evolved after it infected the teenager. Although these findings have inspired multiple alarmist news headlines, there are a couple of reasons for why there is currently no need to panic, in my opinion. First, there are at least two other obstacles that prevent sustained human-to-human transmission of H5N1, in addition to receptor preference. One obstacle involves the H5N1 polymerase enzyme, which normally functions at 40 degrees Celsius, the temperature of the avian digestive tract, but would have to adapt to function at the 33 to 35 degrees Celsius temperature range of the human upper respiratory tract to replicate in a human in order to transmit effectively from this site. Another obstacle relates to the pH inside the endosome, the membrane vesicle that forms around the viral particle once it enters a host cell. H5N1 viruses are only stable in the high pH environment of the avian endosome and would easily degrade in the low pH of the human endosome, leading to inefficient replication and transmission to a new host. Second, scientists have been anticipating an H5N1 pandemic for many decades. There are several effective FDA-approved antiviral drugs that are readily available, and a few experimental vaccines are also in development. The changes observed in the sequence of the viral RNA isolated from the teenager in British Columbia are necessary but not sufficient to trigger a human pandemic. Therefore, the public health risk associated with H5N1 influenza virus is still low at this time. Nevertheless, circulating viruses should be continually monitored for changes in genomic sequences that signal the potential for human-to-human transmission. Meanwhile, try not to touch sick or dead animals in the wild and prevent contact between domesticated and wild animals. In other words, keep your pets indoors and away from wild animals. That's it for today. Thanks again for listening. Please subscribe. My name is Trudy Ray, and I'll see you next time on Catch This.